Chapter 18. We are still at the beginning. So, step by step, the great biologist worked, examining the strange phenomena of birth and life and death. And still there were unsolved problems and mysteries they could not understand. Though they had worked with the brilliance of Aristotle and watched the marvels of nature with the wonder of Marcello Malpighi, though they had studied patiently as William Harvey had studied, there was much that was not yet understood. August Weismann, working in his laboratory at the University of Freiburg in Germany, knew this, and he wanted to add what he could to the work of the biologists who had preceded him. He was particularly interested in heredity. What exactly is it that is passed down from one generation to the next, he asked himself. Why is the son generally like the father? Weismann had been well trained for scientific work. For 23 years, he had studied cell structure at German universities, and he had made some original contributions toward understanding it. But after that, he found, as Malpighi and many others had discovered before him, that work with a microscope puts a great strain on the eyes. In time, he developed glaucoma, a disease of the eye characterized by increased pressure within the eyeball and progressive loss of vision. For 10 years, he could not use his eyes at all. During those years, he says in his autobiography, he listened to music and listened while his wife read aloud to him. Finally, his eyes improved. He was able to go on with his work. He lived and worked till 1914. By that time, he was a white-bearded old man who wore very thick spectacles. The questioning look that had been characteristic of his younger days was gone. He thought he had found the answer to a part, at least, of the problem he had been trying to solve. Quote, the reason the offspring tend to be like the parent, he said, is that it is made of the same stuff. The cells of the body live and die again, but among them there is one that is indestructible. It is the germ cell. It carries life from one generation to the next, and in this sense it is immortal. Unquote. Then he explained it further. He said in substance, quote, when the sperm unites with the ovum to create a new organism, the germ cell is passed down from one generation to another. The body cells develop only to nourish and protect these age-specific life. The germ cell is a specific substance, he went on. It has a definite chemical molecular structure. This is the bearer of heredity. It is passed down through the ages, bringing with it the accumulated characteristics which the new individual inherits. In this sense, the new creature is a chip of the old block. Unquote. Perhaps it was fortunate that Weismann lived when he did, for microscopes were being continually improved, so that cell structures in it had been impossible to see at an earlier time were easily discernible. Moreover, the biologists had found ways of staining a part of a nucleus with a dye that had little effect on the fat, starch, and protein that comprised the main part of it. The part they could stain was called the chromosome, which is a Greek word meaning color body. When Weismann's theory of germinal continuity was announced, therefore, scientists in Germany, Sweden, England, Russia, and the United States and other countries began to investigate the chromosomes, which were the bearers of heredity. They used great ingenuity and patience. As they studied the chromosomes, they soon saw that they were not all exactly alike and that they changed from time to time. They assumed different shapes. Sometimes they looked like rough sections of yarn, sometimes they were curved like sausages, or they might be twisted into irregular V's and W's. The scientists also found that each species had a fixed number of chromosomes. They made a list of them as follows: shark, 36, mouse salamander, trout, and lily, 24, worm called Shigita, 18, some kinds of snails, ox, guinea pig, and man, 45? 48? No. Nope. 46 for people. Grasshopper, 12. The crustacean called Artemia 68, gypsy moth 62, P14, sea urchin 54, 3 kinds of wheat 14, 28, 42 respectively, 3 kinds of fruit flies 8, 10, 12 respectively. So the prying eyes of the biologists were seeing what men had never seen before, but there was more to be discovered. Each separate species they observed had an even number of chromosomes. When male and female are about to unite, the number of chromosomes in egg and sperm were reduced to half the original number. So the multiplication of chromosomes in each species was controlled, and the new individual started life with the right number for its species. Why was this so? The scientists could not tell. They knew, however, that the germ cells of living things were not a fused product. The chromosomes that passed the complex traits of an individual down from one generation to another were a mosaic of particles. These particles were submicroscopic, far too minute to be seen, yet the biologists did not doubt their existence. Each particle, which might be strung like a precious bead on the fragile thread of the chromosome, must be a separate entity, perhaps differing chemically or in some other way from the rest, they believed. They're not wrong. Soon young biologists everywhere were finding out more about these minute particles that determine the character of life. The most distinguished work in the field was soon under way in America. Thomas Hunt Morgan, working at Columbia University in New York, was perhaps the most successful experimenter, though many others did brilliant work. They all had the same purpose. They wanted to prove, if they could, that Mendel's law was universal, and they wanted to find out what brought de Vries mutations about. They wanted, in fact, to understand heredity. At first Morgan had some difficulty in finding a good subject for his experiments. He tried mice, rats, and pigeons. For one reason or another, they were unsatisfactory. Then he tried experiments with plant lice, and decided they were unsatisfactory too. At last he heard of a man at Harvard who had been making some experiments with fruit flies. Someone has said that the fruit fly, Drosophila melanogaster, must have been created especially so that Thomas Hunt Morgan could make experiments. It was the ideal subject for his study. The small insect measures only about a quarter of an inch in length. Most people have seen it often, hovering over overripe peaches, plums, pears, and other fruits. It completes its life cycle from egg to winged adult fly in about 10 days, and its lifespan is about 90 days. It can supply the experimenter 30 generations in a year. It multiplies rapidly so that it is possible to raise 1,000 fruit flies in a milk bottle. It thrives on a diet of bits of banana or other fruit, and apparently has no objection to living in a laboratory. And best of all, it has 4 very large chromosomes that are not hard to see with the microscope. Morgan had some fruit flies sent to his laboratory. They multiplied, and soon he had thousands and thousands of them. He set them in carefully labeled bottles in rows along his shelves. This was in 1909. Then he began to submit his specimens to all sorts of tests. He tried unusually high temperatures and unusually low temperatures. He kept them in the dark and treated them with blazing light. He treated them with acid, and he fed them with strange diets, hoping in some such way to produce a mutation, a fruit fly that was different from the common run of its fellows. Nothing particularly interesting happened. Then, in April 1910, quote, in a pedigreed culture Drosophila which had been running for nearly a year, through the considerable number of generations, a male appeared with white eyes, unquote. This was extraordinary, for all the other thousands of fruit flies in Morgan's bottles had red eyes. Here was mutation that he could use for his experiment. He mated the white-eyed fly with a red-eyed female from a pair, fed the pair some banana, plugged up the bottle with cotton, and watched for the result. The first generation of newcomers had red eyes, for red eyes were apparently dominant. Morgan was greatly satisfied as he waited for the second generation. These, as he expected, were in the proportion of 1/3 red eye, 1/3 white eye, 1/3 mixed red and white eye individuals. I don't know why they were describing it that way. That doesn't make a lot of sense. Mendel's law was working. As time passed, Morgan and his assistants, examining the fruit flies under their microscopes, found 25 distinct mutations. Fruit flies with crooked wings, with speckled wings, with variations in color, without eyes. They interbred and crossbred carefully. Very soon, Morgan had some new facts on heredity. The factors that bear the various herited characters are like little discs strung on the thread of the chromosome, he said. He guessed there might be as many as 10,000 little discs, which he called genes, in some chromosomes. Among these genes are two which he called X and Y. They determine the sex of the new organism. When the egg is fertilized by the sperm, the slender threads of the chromosome twist around one another in what is called chromosomal embrace. I don't know anyone who calls it that. Then some of the genes from one side pass over to the other side. This is why the new organism inherits qualities from both parents and from ancestors on both sides. Some of them might be knocked off or brushed aside or lost. This would be sufficient cause for mutation. But it is the combination of genes that determines the character of the new individual as an organism that is new, for it bears a new combination of genes, yet old, for it is composed of the same material that has been passed down from generation to generation. For several years, Morgan and his assistants worked over their fruit flies, recording their pedigrees and examining them for mutations, observing how some genes were linked so that certain types appeared only in the males and others only in the females. Then they realized the genes appeared in a regular order in the chromosomes. The genes for red eyes always occurred at the top of the chromosome, while that for speckled wings would be near the bottom. Gradually, they succeeded in photographing the genes, and so made a series of chromosome diagrams or maps. How Mendel would have enjoyed seeing those fruit fly experiments! How De Vries would have marveled at the gene maps! But what is the practical importance of work like Morgan's? Will it help us to produce richer grain fields, heavier fruits, sturdier cattle? Will it help to produce a more intelligent or wiser race of men, or a healthier one? Not yet, at any rate. Grain, fruit, and cattle are being improved by selective breeding, but not with the help of gene maps. Very little indeed is definitely known about human heredity. This does not mean that the present studies in heredity will never be of practical use, for whatever leads to a clearer understanding of nature's ways will be useful. 300 years ago, William Harvey announced that he had watched the circulation of the blood in the human body and so encouraged men to set aside their old notions of and investigate the function of the body's organs. In the same way, biologists who study the arrangement of the genes in various species are just at the beginning of a new study. Great though it is, the work of the modern biologist must be considered with humility. There are so many things in the world that cannot yet be explained. All forms of matter on Earth, organic and inorganic, are made up of atoms of different elements, we say. All living things have developed through a long process of evolution. We know something about them. We know something about laws of heredity. But we still do not understand the process by which a green leaf manufactures food. Nor do we know exactly how a migrant bird sets off on its long journey. Neither do we understand why animals and men can remember what is past or why men are able to plan for the future. We have not come to the end of understanding. We are still at the beginning.